Okay, so I'm going to try doing the rest of chapter 17 in this lecture video, and then it is done. Okay, so let's start sharing. I'm not on the correct page. So molecular techniques for analyzing DNA. So first off, okay, this is good. I didn't realize that I hadn't gone over this already. But first off, one of the main ways that we analyze DNA is by looking at its fragment size. So we've seen a lot that we use restriction enzymes, right? We use restriction enzymes, which basically fragment and cut the genomic DNA or whatever DNA we're looking at. They just cut it into small little pieces. And so a lot of times we can basically isolate these little pieces by making them run on a gel electrophoresis, on a gel. So making them carry out a gel electrophoresis. Essentially how this gel works is that we have this polyacrylamides or agarose labyrinth of a gel. So it's very porous, so the DNA fragments can finagle their way through it. And this gel is put in this weird, cool looking apparatus thing where if you have the negative electrode here and the positive electrode here. I honestly thought that the positive electrode was called a cathode. I could have sworn. No, it's called an anode. Okay, never mind. So it goes towards the positive electrode. And this works because DNA is negative, right? So when we apply this electrical current, we have this negative the negative, I can't say it because it, for some reason it sounds wrong to me. The negative electrode at the beginning and then the positive electrode at the end. And our DNA gets basically put into these little wells. So when our DNA gets put into these wells and we apply an electrical current on the gel, essentially what happens is that the DNA starts migrating towards the positive electrode, through the positive electrode. So the DNA starts migrating there. And as it migrates proportional to its molecular weight, meaning the bigger molecular weighted DNA, so the bigger fragments take a longer time to reach the end compared to the smaller little DNA fragments that can go really, really quickly. So it takes into account these physical properties of the DNA, pretty much its size and the charge of the DNA that it's attracted to that positive electrode at the end. So if you see here, this particular band, this is a DNA fragment. This DNA fragment is smaller than this DNA fragment right here. This DNA fragment 2, this third one, is smaller than this second DNA fragment. And it's bigger than this fourth DNA fragment. Okay? You can also basically see the size of the DNA fragments by running your DNA sample alongside a molecular ladder. Molecular ladder is full of fragments of DNA of known sizes. So usually when you load a molecular ladder, you get in a little tube, and on that tube or next to the instructions near the tube tell you which fragments, like all the fragments that you're supposed to get when you run the DNA ladder, and how big each fragment is. So if the DNA ladder says that this, I don't know if you can see, but it's quite faint, this fifth fragment down on lane, I think it's the 6th lane or 7th lane, in lane 7, that's where we put our molecular ladder. This 5th fragment down has a base, has like, I don't know, 500 base pairs. That means that anything on the same plane as this size fragment, so this band right here, which is on the same plane as this fragment, is basically the same size. So it too will be approximately 500 base pairs. Now, there's different kinds of gels that can separate everything, like they can separate DNA fragments even by like 1 base pair in length. It's incredible. Usually we use polyacrylamide gels to do that because they're a little bit more matrix-y. Okay, so this gel electrophoresis is used in conjunction with many different techniques in molecular biology. Another technique that we use in molecular biology is nucleic acid blotting. Actually, before we go there, I believe your textbook talks about restriction mapping, and that's just basically us sequencing and seeing where the restriction sites are within your genome, or within the genome of whatever organism you're looking at, to see where the restriction sites are, because you guys know again how important these restriction enzymes are. The restriction site is just a place where we can then go and cut with the specific restriction enzyme that cuts that specific recognition or restriction site. So we kind of glossing over that because it's not something too important, but just know that you do take the extra step of mapping and finding every single restriction site in the genome. Okay, another one is nucleic acid blotting. Nucleic acid blotting is pretty cool. I've always loved making them because it felt like a little like kid project or something, because you literally have sponges and membranes and like a little weighted brick. Like sometimes we would even use textbooks, don't judge, to like weigh things down. It was pretty awesome. And then like paper towels and all this stuff. And so it looks kind of like a really funky experiment, but in actuality, it's like a real big deal, right? So a southern blot is used to identify which clones in the library contain given DNA sequences and to characterize the size of fragments from restriction digests and determine whether clone contains all or part of a gene. So say you have a DNA sample. So actually, before we go in, there's two components to this southern blotting. You have the separation of the DNA fragments by gel electrophoresis, which is what we just covered. And then you have the hybridization by using labeled probes. And in order to get to that hybridization by using labeled probes, you have to go through a southern blot where you have to go through some funky little weird sponge-weighted thing apparatus. Right, so your samples are of the DNA that you want to probe. So say you have a bunch of DNA fragments and you want to find just a few that are going to stick to your particular probe. Remember, a probe is usually like a single-stranded DNA or a piece of RNA, right? A small little piece of RNA that is complementary to whatever target sequence you're looking at. And usually the probe itself is equipped with like fluorescence, like a specific kind of tag, like fluorescence, or it could even go through like autoradiography with X-ray films. So the sample of the DNA to be probed are cut with restriction enzymes, right? So you have all these different fragments, and then the fragments are separated through electrophoresis. At this point, the DNA is going to have to be denatured, right? So we're going to heat up the DNA, denature it, heat up the DNA in the gel, denature it as well, so that we have single-stranded DNA, because as you guys know, the probe is single-stranded. It's a single-stranded nucleic acid, and it needs another complementary single strand to bind to. So we denature our DNA so that it is no longer double-stranded. It is now separate and single-stranded. And then once we do that, we start looking at, we put the gel in this weird, fun, I keep calling it fun, but I honestly had a blast making these. Sponge wick, this little sponge bath, where the gel is basically put through here, right in the middle. The middle, so you have your buffer, then you have the sponge, your gel, and then your like DNA binding filter. So what your DNA wants. So essentially, we're trying to move the DNA from the gel to this filter, right? And then on top of it, you have a towel, not a towel, sorry, a weight. So what occurs is that through capillary action, the buffer rises through the sponge. It goes through the gel, and as it goes through the gel, it picks up the DNA and then attaches it to the filter found right on top of the DNA. It attaches it to the filter, the DNA binding filter. So let's see how they explain it here, or I explain it in my PowerPoints, which I made through the textbook, so it's fine. Yeah, the gel is then placed on a sponge wick in contact with a buffer solution and covered with a DNA binding filter. Layers of paper towels or blotting paper are placed on top of the filter and held in place with a weight. Capillary action draws the buffer through the gel, transferring the pattern of the DNA fragments from the gel to the filter. It's pretty much what I just said. Sorry. So at this point, the filtered DNA has those single-stranded DNA, because remember, we denatured them, has a single-stranded DNA on it. Okay, so we can then take this filter and place it in a little fun little baggie, right? Fun little heat seal baggie, and we add our radioactive probe or even our fluorescent probe, whatever kind of probe you want, right? We add our probe to this little baggie. We kind of let it, we don't really shake it around, but we let it sit and allow for the probe to find its target sequence. Because again, the probe is a nucleic acid, it's going to bind to a complementary single-stranded sequence. So once the probe gets attached, we then wash the rest of the probes away, anything that hasn't attached, because you see here lots of little probes are there. They're all identical, they're all looking for the same complementary sequence. But if some don't attach because another probe has attached to the DNA prior to it, it's going to get washed away. So as soon as we wash it away, the filter is washed to remove any unbound probe. When dry, the X-ray film is applied for autoradiography, and then you can start seeing any kind of like DNA that was specific to your probe will show up. So out of all of these targets right here, all these fragments right here, sorry, not targets, fragments, all these fragments at the top, we only have these few fragments will actually be visible because they have been hybridized to the probe, and that means that that is your target DNA. So then you can go back to your gel and kind of see and isolate your DNA and all that good stuff. Okay, so the the hybridized fragments show up as bands on the X-ray film. And again, you can also use fluorescence to do this as well, but usually for nucleic acid blotting, it's not. So with the southern blot, you also have things. So this was the first one that was made. The southern blot was to transfer DNA. We also have things like the northern blot. The northern blot is to transfer mRNA or RNA essentially. And then we also have the western blot, which are also quite fun. And the western blot is to transfer proteins. So it's the same kind of technique. You always still allow the nucleic acid or even the protein to be separated through gel electrophoresis, right? And then you just transfer it to a specific filter. Oh, here we go. So northern blot analysis is the RNA blotting. It's used to determine whether the gene is actively being expressed in a given cell or tissue. So we can find a cell, or we can find a given like tissue, or even a cell during a specific time of development or something like that. Like, so specific localization, specific timing, and if I get its mRNA, basically the genes that they have expressed, get their mRNA, allow them to run on a gel and use this, right? And then transfer them over to a filter. So it's used to study patterns of gene expression in embryonic tissue, cancer, and genetic disorders. It also detects alternatively spliced mRNA. Now, however, it's not used as often, and that is true. It's not really used as often because we have reverse transcriptase PCR. Remember, reverse transcriptase PCR allows us to clone even all the DNA that has been made into genes. Sorry, all the DNA that has been made into mRNA, right? So it doesn't just look at genes. It takes your genes and we make them into mRNA. Then that mRNA is made into complementary DNA through the use of reverse transcriptase. And then that complementary DNA is PCR. Yeah. Western blot is still used quite often, and this is used for analysis of proteins. Another thing that we do is FISH, or fluorescent in situ hybridization. This basically involves directly putting in a probe to the tissue or to the cell. So without isolating the DNA or isolating the RNA or isolating the protein first, you could just directly, as the tissue is basically in vivo, right? Or in situ means in tissue. So while the tissue is still living, like in a little plate, we have little tissue cultures, you can just add the fluorescent probe, the fluorescent tag, and see where it glows. So this involves hybridizing the probe directly to the chromosome or RNA without blotting. So blotting is that sponge, that weird sponge buffer thing that we have, without the need of that, because again, you're not isolating anything, you're just putting it directly on the tissue culture. It's carried out with isolated chromosome slides or in situ, or which means in tissue sections or entire organisms. I'm going to put this, in situ means in tissue. It's helpful where embryos are used for various studies in developmental genetics. All right, so you can see here, we have some in situ like hybridization where it went specifically to the centromere, right? Here, this is probably like microtubules, GADPH, it's just a thing. Microtubules were highlighted. So you have different places. You could just put like the, where the DNA, like specific areas of the DNA where it could be expressed. So with the microtubules, as you can realize, probes can also work against proteins, not necessarily nucleic acids, right?